Hello everyone, welcome to another lecture of the series which involves CRISPR-Cas system. So today we will be looking at some of the applications and some of the case studies related to the CRISPR-Cas system. So let's begin with application in medicine. We will be continuing this particular topic from the previous studies. So let's explore how CRISPR is transforming the field of medicine. Think of a CRISPR as a superhero medicine that needs to combat the genetic diseases. So, CRISPR in treating genetic diseases. One of the most promising applications of CRISPR is targeting the genetic diseases. Imagine having a tool that can correct the root cause of illness caused by any faulty gene. For example, we have seen the case of sickle cell anemia, and this is the problem that the entire world is facing today. CRISPR have been used in clinical trials to edit the gene responsible for this particular disease, potentially offering a cure. Similarly, in case of beta thalassemia that we also discussed in the previous class, it is also a very rare but genetic disorder, and till now there is no cure. So, CRISPR-Cas is a very promising system. With the help of this CRISPR-Cas system, we can actually treat these diseases. Now, I will briefly include the clinical trials and its success stories. So, clinical trials are essential steps in bringing CRISPR based treatment from laboratory to real patient. Think of them as testing ground to ensure the safety and effectiveness of these therapies. For example, in the US, first clinical trials using CRISPR to treat a genetic eye disorder called Label congenital amaurosis showed a remarkable success. The patients who were once blinded gained some vision. This is also we studied last time. So, very briefly I am just giving you the perspective of these studies. Another notable success story involves Emma, a young girl born with a rare genetic disorders called as Diamond-Blackfan anemia. The CRISPR was used to correct the genetic mutations in her cell, potentially providing a lifelong cure. This success gives hope to patients with previously incurable genetic diseases, showing that the CRISPR has potential to transform the landscape of medicine. Now, I will directly jump to another application in the field of agriculture. So, let us explore how CRISPR is revolutionizing the field of agriculture. So, this briefly last time we have studied. Now, I will include just the bullet points of these particular topics. So, first topic is crop improvement with some sort of genetic manipulations or genetic modifications. So, the CRISPR-Cas system have been used to modify the yield of these particular plants. Not only yields, but also these are used to increase a particular beneficial part of these particular crops. For example, the golden rice and other BT cotton, all these are very famous example, and there is very recently the brinjal, the BT brinjal is also gained a lot of traction. So, CRISPR is very essential to modify these genes in order to improve the crop production as well as the crop productivity. Now, let us begin with another application in the field of biotechnology. So, now that all around the world a lot of different research is going on. So, let us dive into how CRISPR is shaping the field of biotechnology. Think CRISPR as a powerful tool that unlocks the new frontiers in medicine, industry and beyond that. So, the first example that I will be using today is bioproduction and synthetic biology. CRISPR is playing a pivotal role in production and synthesis and synthetic biology, revolutionizing how we manufacture the valuable molecules and materials. Imagine a molecular factory with a CRISPR as a tool and control system. One exciting example in the field of CRISPR is a microbial fermentation. Scientists are using these engineering methods to perform a genetic manipulation in microorganisms to produce biofuels, chemical and even textiles. So it's like running a microbial into a tiny chemical factories. These CRISPRs is also being employed in synthetic biology to create a custom design organism for specific task. For example, the researchers have engineered the bacteria to detect and clean up environmental pollutants such as biological vacuum cleaners. So in this we will be using some of the examples that involves the cleaning of oceans that have been polluted from the oil and other different pollutants. CRISPR, another example I will be using is CRISPR in drug development. In drug development, CRISPR is a game changer, speeding up the discovery of new treatment and therapies. Think of a CRISPR as a compass guiding through the complex landscape of drug research. Researchers are using CRISPR to create a disease model in the lab. For example, they can replicate a genetic mutations that you can observe in patients with genetic disorders such as cancers. This allows scientists to test the potential drugs more effectively, and one of the major things revolutionizing this field is you don't need the entire organism for this particular purpose. What you need is a small section of those tissues such as liver tissues, heart tissues or skin tissues in order to perform these genetic changes. CRISPR is also used to develop a cutting edge therapies like CAR-T cell therapy for cancer treatment. In CAR-T therapies, the patient's own immune cells are genetically modified using CRISPRs to target and destroy cancer cells. The speed and precision of CRISPR in drug discoveries are upgrading from manual typewriter to a supercomputer. It's accelerating our ability to find new cures and treatment. In summary, the CRISPR's application in biotechnologies are vast. From revolutionizing bioproduction and accelerating drug development, these advancements holds the potential to transform the industries and improve the human health. Now, I will be talking very briefly about CRISPR in basic research. So, let's explore how CRISPR is used in advancing the scientific knowledge. Think of these CRISPRs as a key to unlock the mysteries of genetic codes. In this line, first we will study the role of CRISPR in advancing scientific knowledge. These CRISPRs play a pivotal role in advancing our understanding of biology, and genetics is like a powerful microscope that allows scientists to zoom in or zoom out in the small or really minute details of life itself. One of its primary uses gene functional studies. These scientists can use the techniques such as knockout, and in these techniques a specific gene using CRISPR can target these genes and turn off the production, or you can say the exactly the activation of these genes. So, this helps to uncover the role of these genes in a particular state of that particular organism. For example, the researchers use CRISPR to study the function of BRCA1 gene, which is linked to breast cancer. By knocking out this particular gene in mice, they could better understand the role of this particular gene in cancer development. We will study these gene knockout techniques in much detail now. The CRISPR is like a genetic Swiss army knife in the lab. It allows researchers to perform genetic surgery by precisely altering or deactivating these specific genes. In addition to gene knockout studies, the CRISPR enables knock-in experiment. It's like inserting a newly puzzled pieces into a jigsaw to see how it changes the entire picture. For examples, scientists have used CRISPRs to introduce a specific genetic mutations into organisms to observe its effect. It helps us understanding how these genetic changes contributes to the traits and diseases. There is one specific use also. For example, alteration in gene regulation. For example, in the regulatory region, there are some specific portion of genes which can be altered in order to produce a different concentration of these gene products. So, the CRISPR is widely used in these techniques. The CRISPR's precision in gene editing, it's like having a scalpel for genetic surgery, allowing us to dissect the intricate mechanism of life itself. In summary, this CRISPR is a foundation tool in basic research, helping scientists to unravel the mysteries of these genetics in biology. Its application in genetic function studies, then the knockout experiments are advancing our understanding of the life's building blocks. Now, with having background of these applications, now we will briefly study the challenges and limitations of these CRISPR-Cas system. As we wrap up the explorations of CRISPR, it's essential to acknowledge the challenge and limitations that comes to this incredible technology. Think of these as hurdles on the path of progress. Now, I will discuss very briefly some technical challenges in CRISPR-Cas technology. Now, the CRISPR is a powerful tool but it's not without a technical challenges. One major hurdle is the off-target effect. Now, in our previous classes, we have very briefly discussed about these off-target effects. Today, we will see into much more detailed version. So, imagine if you are trying to paint a picture, but your brush sometimes splatter the paints where you don't want it. So, ensuring the precision of CRISPR is like improving the accuracy of brush stroke. So, now the another challenge is the delivery method. So, getting CRISPR components into the right cell can be just like trying to send a package to a specific address without knowing a postal code. So, this poses a lot of challenges in the CRISPR delivery system, and additionally these ethical considerations are and regulatory frameworks are evolving the challenges. So, balancing scientific progress with ethical responsibilities is like a walking on a tightrope. So, these challenges combinedly give a lot of different scope for researchers like yourself to increase the efficiency and accuracy of these methods. Now, very briefly, we will also see the future prospects of these CRISPR-Cas system. So, despite these challenges, the future of CRISPR-Cas technology is incredibly promising. It's like a new chapter in the book, full of exciting possibilities waiting to be explored. Scientists are working tirelessly to improve the CRISPR precision and reduce the off-target effects. Imagine upgrading your tool set to become more reliable and efficient. Now, I will be discussing some emerging CRISPR technology. So, in brief, these techniques are like prime editing and the base editing, often very more precise and potential for the safe genome editing. Think of them as a next generations of tools. Now, the future prospects also include innovative applications that we haven't imagined yet. CRISPR could play a very important role in curing these diseases very precisely, protecting the endangered species or addressing the environmental challenges. Furthermore, as this scientific community continues to collaborate and develop the guidelines, we can ensure the responsible and ethical uses of CRISPR technology. In summary, while CRISPR has its challenges and its limitations, its future prospects are bright as we have overcome the technical hurdles and navigate the ethical consideration. The CRISPR is poised to continue transforming the science, medicine and our world. Now, I will cover the future directions. As we look to the future, it's fascinating to consider what lies ahead of CRISPR technology, think of it as a peering into a crystal ball to glimpse the innovation on the horizon. Now, what lies ahead of CRISPR technology? The CRISPR technology is on an exciting trajectory. Researchers are continuously pushing the boundaries to make it even more versatile and powerful. One direction is enhancing the precision. Scientists are working to further reduce the off-target effects, ensuring that the CRISPR edits only the intended gene. It's like upgrading from regular scalpel to a laser scalpel for your surgery. Another future direction is expanding the CRISPR's toolbox. Now, new Cas protein and advanced delivery methods are being developed, like discovering new tools into a toolkit to tackle different tasks. CRISPR technology is also likely to become more accessible and cost effective, making it available for more researchers and potentially speeding up these discoveries. Potential game changing applications. The potential game changing applications of CRISPR are boundless. It's like having a magic wand that can reshape the world of science and beyond. Imagine a world where we can edit genes of crops that can withstand the effect of climatic changes, ensuring the food safety for the ever growing populations. Now, consider the possibility of personalized medicine, where treatments are tailored to an individual's unique genetic makeup, offering more effective and less invasive therapies. These CRISPRs could revolutionize the conservation effect by helping to save the endangered species from extinction through genetic rescue and modifications. Furthermore, the space exploration could benefit from the CRISPR's technology. These scientists are exploring how to use it to modify the organisms for long duration space travel and colonization. In summary, the future of CRISPR technologies holds immense promises. With greater precision, expanding tools and endless applications, CRISPR is poised to shape the world in ways where we can only begin to imagine. Furthermore, these scientists are using these technologies to discover the new emerging field on a daily basis. For example, in case of exobiology, some of the scientists, they are trying to collect the bacteria or the living organisms in extreme environmental conditions, such as the caves which is full of sulfuric acid, or in case of exobiology, they are trying to get these organisms in the upper surface of the earth, just like stratospheres and others. And also some of the examples where these techniques have no boundaries is discovering the life on a meteor or other spaces, just as Mars or Moon or other places. So, if we are able to collect the genetic materials from these extreme environmental conditions, we can learn how the genetic makeup of these organisms are, and we can employ those conditions to our existing systems, and these modifications can allow these organisms to grow in these specific but very harsh condition, and they can be helpful in our in our industrial production of the lot of rare material or rare chemicals. Now, in the end part of today's lecture, we will be looking at some specific case studies. These case studies are the examples where scientists are able to effectively treat a particular disease or cure of these particular diseases. So, I will discuss the first case study here. So, in a groundbreaking case study published in New England's Journal of Medicine, the researchers utilized the CRISPR based gene editing approaches on a 27-year-old patient diagnosed with HIV and acute lymphomatic leukemia. The short form of this is ALL. Their aim was to mimic the natural immunity by engineering CCR5 ablated the hematopoietic stem cells and the progenitor cells, known for their resistance to HIV infection due to the absence of CCR5 genes. Despite achieving a successful remission of the patient's leukemia, the edited cells proportion remarked to too low effectively reduced the HIV viral load. The patients underwent the allogenic hematopoietic cell transformation, short form of is allo-HCT, with donor driven CD34 positive hematopoietic stem cells and progenitor cells, which were subjected to CRISPR editing with 17.8% accuracy. While the treatment occurred, the ALL remained in the remission. Only a 5% of CCR4 disruption was observed in lymphocyte, which is low efficiency. This low efficiency was attributed for the competitive engulfment of CD34 depleted cells containing a significant proportion of total CD34 positive cells. Although the gene editing procedure shows no adverse effect or off target mutations in this study, the authors highlight the need for further research to enhance the gene targeting efficiency and expand the safety analysis of CRISPR-Cas9 mediated CCR5 in case of hematopoietic stem cells and progenitor cells. It is important to note that this report was based on a single patient and a single center, limiting the generalization of the findings. In conclusion, this pioneer case study demonstrated the potential of CRISPR based gene editing in treatment of patients with HIV and acute lymphomatic leukemia, while the engineering cells lead to remission of leukemia and displayed a promising outcome to the study. So, this study also revealed the challenges of achieving sufficient CCR5 disruption to actively lower the patient's HIV load. The findings underscore the need for further research to improve the gene editing efficiency and broaden the safety analysis of this innovative approach. Although based on a single patient and a center, this study opens a new avenue for explorations in the field of gene therapy and personalized medicine. We will be looking at another case study. So, gefitinib and erlotinib are the drugs that are used to treat a type of lung cancer called as non-small cell lung cancer, shortly abbreviated as NSCLC. They work by targeting a particular protein called as epidermal growth factor receptor, also abbreviated as EGFRs. However, sometimes these drugs stopped working because of a challenge in EGFR called as T790M mutation. These mutations makes it harder for a drug to work. To solve this problem, a company called as AstraZeneca created a new drug called as osimertinib. It was designed to target T790M mutation, but in some cases even this drug stopped working because of another mutation called as C797S mutation. To find new treatments quickly and save money, the scientist uses a powerful tool, just what we are learning today, called as CRISPR-Cas technology. They studied it to create the C797S mutation in cancer cells in the laboratory scenario. Then they tested different drugs on these cells to find the ones that could treat these new mutations. This method helped them to discover a potential medicine and treat these diseases in a faster and more efficient way. Now, we will look at another case study. So in this case study they have used macrophages. So macrophages are type of immune cells that helps the body to respond the injuries and infections in people with lung diseases called as chronic obstructive pulmonary diseases COPD. These macrophages don't work well to reduce the inflammations. Scientists believe that a protein called as salt induced kinases, shortly abbreviated as SIK, might be a good target for the treatment of COPD. These studies by a company called as Merck found out that if they block SIK protein, it changes how macrophages behave. They become more inflammatory, which is a good for COPD patients. SIK comes in 3 different forms in the body and scientists wants to figure out which one to target. They use a powerful tool called as CRISPR-Cas to create the changes in these forms in a lab grown cell. These alterations are acted as the real macrophages, and when they tested them against these particular diseases, they found that the right combination of SIK forms that could reduce the inflammations. This discovery helps scientists to move closer to finding a new treatment for COPD. Now, I will also introduce last case study. In 2014, some scientific studies suggested that a protein called as Mu-T-homolog 1, also shortly abbreviated as MTH1, could be a good target for cancer treatment. However, the researchers at the AstraZeneca company wanted to double check this idea. So, they uses CRISPR-Cas9 modifications and they modify the MTH1 gene in the cell. Basically, they made changes in this gene so that it could not make the MTH1 protein anymore. So, it is like a knockout technique. So, they found that the cells without MTH1 protein grew just as a normal cells. Then they treated these modified cells with a specific drug that was supposed to affect the MTH1. Surprisingly, the drug had no effect or no different effect in these modified cells compared to the normal ones. It means that MTH1 might not be a good target for cancer treatment at all. This discovery saved a lot of time and a lot of resources and avoid the pursuit of the wrong target. So, I will summarize these 3 topics that we discussed in the context of CRISPR-Cas technology. The possibilities are endless. At the same time, there are ethical considerations that needs to be followed. Along with ethical consideration, there are a lot of challenges which required a further studies in this direction, and I believe tomorrow the scientists will be able to find a newer approach in this particular field. So, this is all from my side. Thank you very much. Happy learning.